Silantes, stable isotope-labeled biomolecules. With this focus, we have been producing isotope-labeled fine chemicals for applications in NMR and mass spectrometry for over 20 years. Our history began as a spin-off of the Max Planck Institute of Biochemistry in Munich, with a patent on a technology for biotechnological fermentations. Even today, this concept sets us apart from the competition. All of our products come from a cascade of biotechnological fermentations without using chemical synthesis routes. In this talk, I would like to give you an insight into our technologies and products, but more importantly, I would like to show you the interesting results our customers achieve with our products and how we can contribute to the success of your research project. By way of introduction, I would like to start with our core competence, the fermentation of the bacterium known as Cupria vidus necator or Ralstonia eutropha. Ralstonia gained its general importance in the industry as a producer of polyhydroxyalkanoates or PHA, a biopolymer that is biodegradable and used to produce bio-based plastics. However, for Silantes, the most important aspect of Ralstonia is that this bacterium can change its metabolic pathways according to the substrates available. If an organic carbon source is available, Ralstonia is capable of growing heterotrophically. However, if no organic carbon is available, Ralstonia can also grow autotrophically and utilize inorganic carbon. This attribute is called facultative chemolithoautotrophic. Silantes uses both the autotrophic and the heterotrophic metabolism of Ralstonia. As raw materials, we use inorganic stable isotopes such as 15N ammonium chloride, 13C carbon dioxide or deuterium oxide. In the heterotrophic fermentation of Ralstonia, we label the biomass with the stable isotope of nitrogen on a large scale in our 100-liter bioreactor. The heterotrophic fermentation is not suitable for labeling the biomass with the stable isotope of carbon or hydrogen, as it would be necessary to introduce isotopically labeled glucose as an organic carbon source. Labeled glucose, however, is relatively cost-intensive and would make the fermentation uneconomically. If the biomass is to be additionally 13C or deuterium labeled, the autotrophic metabolism comes into action. Here, the characteristic is used that 13C can be introduced in the inorganic form of 13CO2. This makes the fermentation economical, as carbon dioxide is the most cost-effective source of 13C. In the autotrophic fermentation, Ralstonia performs the oxyhydrogen reaction to gain energy. This reaction is an exothermic oxidation. If the volume content of hydrogen is greater than 4%, the hydrogen would be converted to H2O with the oxygen in an explosive reaction. Therefore, the volume content of hydrogen must be kept strictly under 4%. To allow this process to be used industrially, we had to develop explosion-proof fermenters and prove the explosion protection under real conditions. In the clip, you can see what happens when the oxyhydrogen reaction takes place in a glass fermenter with a water bath. Under these circumstances, the autotrophic fermentation of Ralstonia would obviously not be safe. Therefore, we've developed an explosion-proof fermenter as shown in the next clip. The explosion taking place in the fermenter can be sensed because of the movement of the hose. With this explosion-proof fermenter, we've obtained permission for industrial use of this process. As a further autotrophic production organism besides bacteria, we use green algae. The most important element in the metabolism of green algae is photosynthesis. Through photosynthesis, organisms are able to build organic compounds with water, CO2, minerals and light alone. Special reactors called photobioreactors are used to cultivate green algae. These provide the organisms with optimized light. Classically, Either plate or tube bioreactors are used for the production of algae in the industrial environment. To meet our specific requirements at Silantes, we have developed our own photobioreactor, which is neither plate nor tube. We call it the Green Compact. As shown in the clip, the Green Compact consists of a horizontal glass cylinder. The lighting is provided by fluorescent tubes in glass pipes that run through the entire interior. Compared to commercially available plate or tube bioreactors, the innovative design of the Green Compact has an outstanding advantage. Due to the illumination from the inside, the Silantes bioreactor, as the name also reveals, is more compact than a tube or a plate bioreactor. This results in an optimized space-time yield, increased robustness and stability, and improved ergonomics. The stable isotope-labeled biomass from the various fermentations is introduced as an organic carbon source in a second cascade step into another heterotrophic fermentation to extract further complex biomolecules. With these different upstream technologies, we label biomass by metabolomic labeling with the stable isotopes. We also make our know-how and fermentation technologies available as a service if one of your projects requires specific labeled biomasses. In our downstream process, all biomass is made usable, the zero waste concept. A stable isotope-labeled cell growth medium for yeast and E. coli cells is produced from the hydrolysate of the biomass. With this medium, stable isotope-labeled proteins can be expressed highly efficiently. Furthermore, stable isotope-labeled ubiquitin is purified, which can be used as a ready-to-use NMR reference standard. All components of the RNA and DNA are also purified. These are available as uniform or site-specific isotope-labeled nucleotides or also as phosphoramidites. Alternatively, we also offer the service of synthesizing RNA and DNA sequences as desired up to a length of more than 70 bases. We also purify stable isotope-labeled amino acids. For the application in the field of proteomics, we offer all necessary media for a Silac experiment in addition to the essential Silac amino acids lysine and arginine. For both proteomics and metabolomics studies, we offer diets for various model organisms such as mice and flies. These are available with either uniform or amino acid specific labelings. In addition to this, we also offer already isotopically labeled tissue of the model organisms. Besides production, we also try to further develop our products and services. In some cases, this is done in close cooperation with research groups worldwide. As an example, Andre Simpson's research group from the University of Toronto is studying the effects of exposure on organisms in vivo under the title In-vivo NMR, bridging the gap between contamination and environmental and human health. In this type of experiment, organisms are kept alive in the NMR device. This allows real-time monitoring of changes in metabolites. Using living 13C enriched Daphnia, anoxic stress responses are monitored to identify metabolites. These Daphnia are fed with highly enriched 13C algal biomass derived from Silantes. In this clip, you can see the Daphnia being fed with the 13C labeled biomass in the NMR tube. Thus, the living organism becomes the ultimate biosensor, responding to its environment in real time, while the spectrometer interprets the biochemical changes and provides information on sublethal toxicity. Besides the collaboration with the Andre Simpson lab, we also work very closely together with Harald Schwalbe's group. The Schwalbe Group is the leading bio-NMR group at the Johann Wolfgang Goethe University in Frankfurt. Due to recent pandemic developments, the COVID-19 NMR Consortium was founded under the coordination of Harald Schwalbe and his team. Here, scientists from all over the world are working together in a unique project to study SARS CoV-2 using NMR spectroscopy based on the principles of open science. The aim of the project is to determine RNA and protein structures of SARS CoV-2 and to investigate the trackability of those structures by small molecules. Harald's group synthesized isotopically labeled RNA sequences of the virus using uniformly 15N and 13C15N labeled ribonucleotide triphosphates of Silantes. This made it possible to calculate the structure of the sequence based on NMR measurements. The illustration shows a structural bundle from the SARS CoV-2 5' untranslated region. The structural bundle is 27 nucleotides long and folds in a secondary stem loop structure. This stem loop, the so-called 5SL1, ensures the translation of the viral RNA. This RNA sequence is especially interesting in connection with the viral non-structural protein 1 or NSP1. The protein is used by SARS CoV-2 to paralyze the host cell's immune defense by shutting down the translation of most of the host cell's mRNAs. In the end, the Schwalbe group was able to help clarify the structure and function of the SARS CoV-2 RNA using the stable isotope labeled rNTPs from Silantes. We are honored to be a part of the valuable research of André Simpson, of Harald Schwalbe, and of many other research groups. We look forward to the future and to contributing our experience and products to the success of your projects in both bio-NMR and mass spectrometry.